Third Eye Weekly. Joining us today is Ksenia Gnedeva, postdoctoral associate at the Laboratory of Sensory Neuroscience at Rockefeller University. She's here to speak with us about her latest research into locating proteins inside the ear that can possibly produce new hair cells and correct hearing loss for the future. Thanks for joining us.
Hi guys, thank you for inviting me.
Great. So, let's start out with what first got you interested in this line of study?
Right. So as PhD, I was focusing more on skin biology, but my background is embryology and developmental biology. And I got interested in sensory neuroscience and especially hearing because it's a very tricky subject. I don't think that many people know that mammals are the only species that cannot regenerate their hearing when they lose them. Pretty much, all other vertebrates, including birds, amphibian, and reptiles, when they lose their hearing due to, loud noise exposure or any other way, they would rapidly regenerate their hair cells and therefore restore their hearing and balance. And mammalian species are the only ones who cannot do it. And so that's what got me interested in this subject, just because there's not enough research on it.
Interesting. So along these same lines, maybe tell us a little bit about how these hair cells in the ear are important to maintain balance in hearing.
All right, so hair cells are the sensory receptor cells that are present in all of the sensory organs of the inner ear. They are present in organ of Corti. This is the organ that allows us to hear. And in vestibular organs like utricle, sacculus, and the crista of semicircular canals. These cells are very similar in their morphology, and they have this funky looking organelle called hair bundle on their apical surface. And so this hair sticking out of the surface of the cell, when you introduce a shearing motion due to sound exposure or balance, those hairs, they slide against each other, and therefore the distance between the tips of those hairs increases. And that's how the cells activate the transduction channels on top of the stereocilia and allow us to hear, for example. So in mammalian tissue, or in actually any other sensory tissue, the cells are also surrounded by another type of cell that is called supporting cells. And the supporting cells allow hair cells to function properly, and also in non-mammals, they are able to differentiate into hair cells when hair cells are lost. So yeah, you can think of hair cells as a sensory receptors of the inner ear.
And so, what are some of the most common hair loss, reasons for hair loss in mammals?
Yeah, so unfortunately, living in New York City is not the best thing you can do. So the loud noise exposure is probably the most common reason for, on a daily basis for everybody. Amongst top reasons are also use of certain antibiotics. They're called aminoglycosides, and unfortunately, some countries still commonly use those antibiotics. Also, it can be a simple ear infection that will cause hearing loss. There are a lot of genetic conditions that cause that as well. And probably, also one of the most common reasons is just aging. It's sort of interesting. We only have a few thousand hair cells, and it might sound like a lot, but for a person who works in skin biology before, I just got a few thousand cells of my surface of my skin, and this is pretty much all of the hair cells we've got. So we're born with all of the hair cells we will ever get, and we gradually lose them due to all those reasons that I mentioned before. So yeah.
So how did you and your team go about examining and identifying some of the possible genes that could help in the reproduction of these vital hair cells?
Right. So what we decided to do was rather a simple experiment. It's, in order to understand how to restore something, you need to understand first how it forms in the first place. And that's what we decided to do. We decided to look at the genes that were important for development of the hair cells in the mammalian vestibular organ in the first place. And we did that using a technique called RNA sequencing. So you can just look at all of the genes that are expressed in the tissue at a certain time point. And then we looked at them early during development, and then when the utricle was developed completely, and then looked at what genes changed their expression levels. And that's how we found this family of transcription factors called SOC-C genes that were dramatically downregulated with age. So as soon as animal stops forming hair cells, the expression of those genes dropped drastically.
So once you were able to identify those genes, SOX4 and SOX11, correct?
Yeah, that's correct.
How did you then be able to turn it on and off in order to see if it could be regenerated?
Right, so there are a lot of genetic tools available, and to turn the gene off, we used a Cre-Lox-P system. It's a interesting enzyme-based system that is found in bacteria. The Cre is a recombinase, it's an enzyme that recognizes a certain sequence in the DNA and cuts it. So what you can do, you can put this specific sequence around the gene that you want to get rid of. And then when Cre recombinase will be introduced, it will really just cut the gene out. And so we used that genetic tool to get rid of SOX4 and SOX11 specifically in the inner ear. And when we did that, we saw that ears were, really malformed. The sensory epithelium did not develop properly, and the hair cells were just completely gone. What was also interesting is that even when we deleted only one allele for each gene, so we got rid of only half of the amount of protein in the animals, usually it doesn't lead to any kind of phenotype because animals is able to restore the function with just half amount of the protein. But even those animals have very strong vestibular phenotypes that were very obvious. They had this, circling behavior, loss of balance, and they just couldn't perform certain balance tasks as their wild-type litter mates would. And then, so in order to overexpress the genes, to restore basically the expression of those genes in adult animals, we used another trick, and we just used an adenoviral genome. And we all know that adenoviruses are really good at infecting mammalian cells. We all get cold, we all get flu. And so we used those viruses and just introduced the sequence for SOX4 and SOX11 genes into their genome. So this way, you have a perfect delivery system already, right? You have this adenovirus that is able to infect the cell, but now it's also carrying the protein that you want to introduce into mammalian cell. And so this is how we restored the expression of SOX4 and SOX11 in adult sensory epithelia of mice. And we were surprised to see that it caused a restoration of hair cells. So we saw some new hair cells forming in those adult sensory epithelia. So, yep.
So the experiment worked for the mice, but are there any reasons you can think of why maybe this would or wouldn't work in a human-to-human scenario?
Exactly. So it's always, so what I'm doing at this point, what our lab is focusing on is more of a basic research, right? So we're looked at development of the sensory epithelia in mice. And you can think about it as, in order to write a book, you need to learn the alphabet first. You need to study the language, you need to understand the grammar. So that's what we're trying to do now. But we do plan to try to look at the role of the same genes in the inner ear development of humans. It's of course very tricky to do because the hair cells develop when, in human fetus, really, not in the babies. So what we decided to do, we could use a human embryonic stem cells and then direct them towards differentiation of the inner ear sensory epithelium, and then look at the function of those genes in this tissue. This way, we can see if they're relevant in human inner ear development, and that will give us a hint of whether or not that can potentially work for humans as well.
Great. Well, those are all the questions we have today. Thank you so much for taking the time to speak with us.
Sure, it was very nice meeting you guys, and yeah, thank you.
Likewise, thanks.